Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into this fascinating deep dive, a quick note. If you love what we do here, please take a moment right now to like, subscribe, and share this with your friends and family. That simple act really supports the Sand Science Experience and allows us to keep bringing you these cutting-edge insights. Today, we are undertaking a deep dive into the genetics of illness, but with a surprising twist. We're not just looking at how genes make you get sick, that's, susceptibility, which GWAS has gotten pretty good at. We're asking, what's the genetic difference between getting a disease and actually surviving it? Progression, mortality, that side of things.
It's a really fundamental question, actually, and one that has, well, massive clinical implications. For years, genome-wide association studies, GWAS, they've beautifully mapped the risk factors that lead to diagnosis, lots of success there. But once a patient crosses that threshold, once they're diagnosed, the genetic map for progression or mortality, it suddenly goes, well, pretty blank.
Right. And as the sources we looked at highlight, this blind spot is critical. We spend billions on drug discovery aimed at improving patient outcomes after diagnosis, curing, not just preventing. If we don't understand the genetics dictating who lives and who succumbs to the disease, we're potentially missing key targets that could revolutionize treatment.
Exactly. The researchers behind this work took I think, a brilliant approach. They systematically analyzed data across 7 major biobanks, including massive data sets like UK Biobank and FinGen. Really comprehensive, they focused on 9 common diseases, everything from, say, heart failure and prostate cancer to chronic kidney disease. And their mission was to compare the genetic architecture. Is the gene that causes susceptibility the same gene that dictates death or survival? They used disease-specific mortality as the clearest measure of progression they could get from these data sets.
Okay, let's unpack this then, starting with the building blocks. Yeah. Single genetic variants. We probably walk into this assuming there must be a huge overlap, right? If a variant makes you, say, 5 times more likely to get type 2 diabetes, it seems intuitive it must also make your TTD worse, maybe speed up complications or mortality. Did the data confirm that simple sort of intuitive assumption?
No, not at all, actually. It provided perhaps the most stunning refutation of that assumption possible. The similarity in genetic effects between susceptibility and mortality was, well, it was negligible, minimal. So the researchers started with a validated list, 804 established genetic variants. These are strongly, robustly associated with susceptibility for these 9 diseases. Known risk factors. When they checked those 804 variants against disease-specific mortality in diagnosed patients, the results were pretty definitive. 0 of them were substantially associated with mortality after correcting for the rigorous statistical bar needed for genome-wide significance.
0. Just to be really clear here.
Yeah.
These are 804 pieces of DNA evidence that we know change your likelihood of getting diagnosed. But they have no significant impact on whether you survive the illness once you have it. That's a paradox right out of the gate.
It gets even more counterintuitive when you look at the direction of the effect. A slightly different question they asked. Yeah. For those 804 variants, okay, if a variant increased your risk of getting the disease, did it also tend to increase your risk of dying from it? They found the direction of effect was the same in only 49% of cases.
Wait, 49%. Let's pause there, because that number, it sounds like the statistical centerpiece of this entire finding, doesn't it?
It really is. The researchers calculated the p-value for that overlap, and it was p.5028, which means the observed concordance between risk and outcome was statistically indistinguishable from a coin flip, just random. It's not just that the genetic signals were weak for mortality, it's that they were randomly associated with progression, basically.
That fundamentally dismantles the idea, doesn't it? The idea that genetics provides a linear roadmap from risk to outcome. We sort of assumed the same genes were working through the entire disease process.
Yeah.
This data says they might stop working or at least stop mattering in the same way, maybe the moment the disease is diagnosed.
That's certainly the interpretation they strongly favor. But there was one single striking exception to this lack of overlap. And actually, it kind of reinforces the disconnect. They identified one specific locus, RS7360523, near the SULF2 gene. This one was genome-wide significant for mortality, specifically in patients who already had heart failure. It was strongly linked to poor survival.
Okay, so a clear, powerful mortality signal there for heart failure patients. Found one. Did that same locus show a strong effect on heart failure susceptibility? On getting heart failure in the first place?
No, and this is crucial. That precise variant showed essentially 0 comparable effect on heart failure susceptibility. Its p-value for the risk of diagnosis was 0.87, as close to statistically random, noise as you can get.
Yeah. Wow. So we have a variant that acts like a lethality switch for heart failure, but it seems to play no role in turning on the disease initially. That really does suggest two completely separate biological machineries might be operating here. One that governs the onset of the disease, and a different one that governs the body's ability to survive it once it starts. Okay, now let's scale it from single variants to the bigger picture. Polygenic scores or PGSs. Since most common diseases are highly polygenic, caused by tiny cumulative effects of hundreds or thousands of genes, we use PGSs to summarize that total genetic risk for a patient. Given the failure we just discussed with single variants, did these more comprehensive PGSs for risk perform any better at predicting survival?
Well, they worked perfectly for their intended purpose, predicting susceptibility. That part was fine. For instance, the prostate cancer susceptibility PGS was strongly associated with the risk of diagnosis. It showed a hazard ratio, or HR, up to 1.90 for risk. So yes, they accurately identify who is genetically susceptible to getting the disease. But then, when they applied those same highly successful susceptibility PGSs to predicting disease-specific mortality in people already diagnosed, the scores were weak, or, in most cases, entirely unassociated with survival. The signal just wasn't there.
Can you give the clearest example of that dramatic drop-off in predictive power?
Yeah, look at breast cancer. The established susceptibility PGS for breast cancer, a really well-refined risk predictor, it showed virtually no association with breast cancer mortality. The hazard ratio was 0.98. Remember, if the HR is 1.0, there's zero association.
Right.
So at 0.98, it's essentially flat. The score for getting the disease just couldn't predict dying from it.
That seems like a massive waste of potentially valuable clinical data, if we've been assuming otherwise. But I noticed in the sources that for some diseases, the susceptibility PGS actually trended towards being protective against mortality. How does that work?
Yeah, that's where it gets conceptually interesting and a bit tricky. For chronic kidney disease and prostate cancer, the PGSs trended toward a protective effect on mortality. It seems entirely counterintuitive, right? The genes that make you prone to getting the disease also somehow help you survive it longer.
Exactly. If the genetics that increase the risk of disease actually protect you from dying, doesn't that fundamentally undermine our concept of bad genes? Is there an error there, or is there some robust biological explanation for this apparent paradox?
The researchers tackled this head-on. They considered a potential methodological flaw known as index event bias.
Okay, for our listeners, can you maybe break down what index event bias is and why it matters in this context?
Sure. Index event bias. It can happen when you study genetic effects only after the disease has already occurred, like in these mortality studies. Think of it like this. To be included in the mortality study group, you first had to survive long enough to get the diagnosis and be enrolled. Now, if you have a really aggressive form of the disease and high genetic risk, you might unfortunately die before being formally diagnosed or enrolled in the study. This could potentially skew the remaining patient population, the ones in the study, towards those who are genetically susceptible, but perhaps whose disease form is inherently less aggressive.
I see. So the bias suggests that the population surviving long enough to be studied might have already filtered out the combinations of genes that cause both high risk and very rapid lethal progression.
Precisely. It's a known issue in these kinds of studies. The researchers accounted for this bias using extensive simulations. But their conclusion was that while index event bias might explain some of the weak association, or maybe those protective trends, it does not fully explain the really low concordance overall. They concluded that the weak signal is more fundamental. It's likely due to the low heritability of progression itself. Genetics just doesn't explain much of the survival variation.
Fascinating. Okay, so if the disease-specific genetics, the ones that cause the initial diagnosis, are largely, useless at predicting how long a patient will survive, what does predict survival then? Where should precision medicine maybe pivot its focus?
Yeah, this is, I think, the revolutionary takeaway from the study. If disease-specific genes fail, maybe we need to look for broader, more general factors related to human robustness and health maintenance, general resilience. So they tested a polygenic score, but this one was derived not from a specific disease, GWAS, but from GWAS focused on general lifespan or longevity in the overall population.
Like a general good genes for long life score, not tied to any one illness.
Exactly that. And the results here were decisive. Very different. This general longevity PGS was substantially associated with disease-specific mortality for 7 of the 9 diseases they tested.
Wow. Okay, let me get this straight. In 7 out of 9 common diseases, the general longevity score, just a measure of overall genetic resilience, outperformed the specific disease risk score when predicting survival after diagnosis. So that means Say you're diagnosed with heart failure. Your ability to survive that diagnosis seems to be dictated more by your overall genetic hardiness than by the specific genetic path that led you to heart failure in the first place.
It certainly looks that way based on this data. It's a huge potential shift in thinking. Maybe we shouldn't be looking so hard for the specific genetic flaw in the engine block, so to speak. Perhaps we should be checking the overall condition of the chassis and the tires, the general system. Survival seems influenced more heavily by these general factors related to sustaining life, factors that might apply regardless of the specific diagnosis you have.
And this pattern was further reinforced when they looked beyond just mortality, right? They explored progression using more refined clinical endpoints, specifically in type 2 diabetes.
That's correct. For T2D, they didn't just look at death. They examined progression defined by things doctors actually track. Macrovascular complications like stroke or coronary artery disease, and also microvascular complications, more nuanced measures.
Okay, and what did the sources show when they applied these generalized genetic scores to those specific T2D complications?
They found that PGSs for general cardiovascular conditions, scores derived from studying heart health in the non-diabetic population, were significantly stronger predictors of T2D macrovascular complications than the T2D susceptibility PGS itself. The general cardiovascular score had a hazard ratio of 1.19 compared to the T2D susceptibility PGS, which had a much weaker HR of just 1.04, basically flat again.
It's the same story repeated, isn't it? General system resilience, in this case cardiovascular resilience, seems a better predictor of outcome within a specific disease than the genetics of the diagnosis itself.
Yes. And if we connect this to the bigger picture, it implies the entire biological framework governing the severity and outcome of diseases. It appears largely distinct from the mechanisms governing their onset. Two different plays, maybe.
So why? Why are these genetic influences so profoundly distinct? What are the factors that the researchers think create this massive disconnect?
Well, several issues stand out, primarily related to the environment and measurement. First, environmental factors likely have a disproportionate impact on progression compared to susceptibility. Think about the variability in treatment choices once someone is diagnosed. The quality of hospital care they receive, medication adherence, socioeconomic status, all these things kick in after diagnosis. These external factors might introduce so much noise and heterogeneity into the patient group that the underlying genetic signal for progression just gets drowned out or severely limited.
Right. So essentially, once the doctor gets involved, once treatment starts, maybe the genetics of survival take a backseat to the environment, whereas the genetics of risk operate largely independent of those specific treatment factors right up until the point of diagnosis.
Precisely. That's one major hypothesis. Second, there is a measurement challenge. Disease-specific mortality is widely available data, which is why they used it, but the researchers acknowledge it might be a relatively poor proxy for the true underlying biological progression. It's a very blunt binary outcome, live or die, and might not capture the more nuanced biology of how a disease worsens over time. We might need better measures of progression itself.
Okay, and let's focus on the third point, which you touched on earlier.
Yeah.
The power challenge versus maybe the fundamental issue of low heritability. Is the genetic signal for progression there, but we just can't see it yet because the studies are too small? Or is the signal fundamentally weak, maybe even close to non-existent?
Yeah, that's a critical distinction, especially for deciding where to put research funding next, right? The researchers did conduct an interesting experiment to probe this. They equalized the statistical power, essentially the effective sample sizes, between susceptibility GWAS and mortality GWAS for the same diseases. Even with equal power, the susceptibility GWAS still found 33 significant genetic loci across 5 diseases, while the mortality GWAS found 0 in that comparable power-matched experiment. This asymmetry strongly suggests that GWAS for progression has a much lower signal-to-noise ratio. It implies that progression isn't just underpowered in current studies, it's heritability. The actual proportion of variation in outcome explained by genetics is likely fundamentally low. The genes simply may not explain much of the outcome variation, meaning that just making sample sizes larger and larger might only yield marginal gains. The signal itself is weak.
Okay, so wrapping this up, what does this all mean for the learner, for the future of medicine, and for clinical strategy? What's the big takeaway?
Well, the core conclusion seems pretty definitive from this work. The genetics of risk getting the disease appear largely distinct from the genetics of outcome surviving it. Clinically, I think the immediate implication is we must stop assuming that a good susceptibility PGS will automatically be a good outcome predictor. They seem to be measuring different things. And looking forward, Perhaps the most practical path for predicting patient survival might not be trying to find the specific progression gene for each individual disease. That might be a very hard road. Instead, research efforts might be better pivoted toward understanding and adapting these general genetic signals from traits like overall lifespan or general cardiovascular risk in the wider population, and then applying those more general resilience scores to diagnose patient cohorts. Survival, it seems, might be less about the specific disease mechanism that got you sick and far more about your general underlying biological resilience.
That's a powerful final thought, a really complete conceptual shift, potentially, in where we focus our genetic searchlights for improving outcomes. Thank you for walking us through these truly illuminating, and I have to say, quite surprising findings. This research really does fundamentally change our understanding of the roadmap from diagnosis to outcome. And to you, the listener, thank you for joining us on this deep dive into the genetics of survival. If you enjoyed this journey and found these insights valuable, please take that moment now to leave us a rating, subscribe for future deep dives, and share this with your friends and family. It really does support the Science experience. We'll catch you on the next one.